great to be here, and yeah, I'm very happy to have the opportunity to share the approach that we're taking in Rotterdam to develop personalized, repurposed treatment for GBM, glioblastoma. So glioblastoma is a very aggressive primary brain tumor, which has a median survival of only 12 to 18 months, and that's despite maximal treatment consisting of surgery, radiotherapy, and temozolomide chemotherapy. The factors that make this tumor so difficult to treat are its infiltrative growth into the surrounding normal brain, the fact that it's located behind partially behind the blood-brain barrier, which of course restricts many drugs from reaching this tumor. And then there's the problem of the intra and intertumoral heterogeneity. So I'm a basic scientist. I work in the lab. So we try to represent this heterogeneity in our model systems, which means that we only use patient-derived tumor tissue. And from this tissue that comes in almost on a weekly basis, we derive cell cultures, but also organotypic spheroids. And we can biobank them, and we can use them again for drug screening. If you look at these cell cultures under just a general light microscope, the first thing you notice is that there's a the reported heterogeneity is actually also visible at a morphological level. And that to us raised the question, does that also translate to variability in sensitivity to drugs? And if so, would that be predictive for how the patient would respond if you would be treating with that drug? Well, there's only one agent for which we could at this moment investigate that, the standard of care drug temozolomide. And indeed, as you can see on this graph here, that there is a very large variability in how these cells between different patients, derived from different patients, respond to this treatment. What we did is we divided the group into three groups called responder, non-responder, and an intermediate group. And then we went and looked at how the patients from which these tumors were derived had responded to standard of care therapy. And indeed, the responder cells in vitro are from the patients that had the longest survival, and vice versa for the non-responders. So with this in mind, we thought, well, maybe we can further develop this tool towards a tool to do personalized medicine and find the best drug for a specific patient. But it also raised another question. Have we maybe in past clinical trials in glioblastoma wrongly ruled out all these promising drugs because we have been evaluating them in unselected populations? And that's actually how we became interested in drug repurposing, in the hope that we could identify those drugs that had been wrongly discarded. So we started screening FDA-approved anti-cancer agents. That's been done now on more than 40 glioblastoma cell cultures. And we calculate IC50 value. So yeah, the amount of drug needed to reduce viability by 50%. It's a very large data set. And here there's a small snapshot of a small part of that data set. And what you can see is that actually for each and every drug along the tested cell cultures, you can find both red and green boxes representing the IC50, color-coded IC50 values. And you can see that the difference between red and green is at least 100-fold. So these are very large differences for virtually any drug that we test among a panel of cell cultures. So from here, we would like to move forward and select drugs that we will move towards a screen-based therapy for these patients. And then the difficult part started. How are we going to select drugs from this tremendous database? Because there's so much that you need to take into account. It's not only the efficacy that we see in vitro, but it's also what is the likelihood of this drug passing the blood-brain barrier? How much protein binding does it have? Does it bind to efflux pumps? And then there's a lot of clinical data, hopefully also available, that can tell us something about any reported responses that have been described, safety, toxicity profiles, PK, PD data, and whether the drug is available. All this information is relevant before we can proceed towards a step closer towards this personalized trial. So in our lab, with our team, we're developing tools that can collect all this information and integrate it in more efficient ways. And what we hope is that that has led to this roadmap where we will be selecting drugs based on the ranking that we made. So we prioritize drugs that go into our phase 0 or so-called window of opportunity trial. And in a window of opportunity trial, patients that come in with a with a glioblastoma, they will be put on one of the selected drug candidates from our selection process. They will for 1 week, and then the standard of care, the regular resection will take place. And then we can measure in that tumor how much of the drug actually accumulated in the tumor, and we can compare those concentrations to what is needed to kill tumor cells. And the drugs that pass that test, they will be put on our drug list. And what we hope is that eventually we will be able to set up as soon as we have more than 10 drugs on that list to move forward toward the personalized trial to actually test in patients. And that's where I'd like to end. Thank you.